Hello everybody and welcome to this webinar. My name is Zach Letterhose. I am your host and I'm delighted to be presenting this edition of the Hills USA Educational Bite series. This is all about poop today. It's all about the microbiome of the gut, and we're being led through that journey by Dr. Jen McClay, and we'll introduce her a little bit later on. So without further ado, I'd like to start the presentation. So welcome to everybody. I'd like to officially welcome you to the Hills Pet Nutrition USA Educational Bite series. This is on the microbiome, led by Dr. Jen McClay. To introduce Dr. McClay, I have Dr. Kristen Woolner from Hills Pet Nutrition USA. So, Dr. Kristen, would you like to introduce our wonderful speaker and tell us a little bit about Hills and their mission and why we're doing this webinar series?
Absolutely. So first of all, thank you all so much for joining us. For those of you who are in the U.S., it's likely a little bit of later night learning, and so we really appreciate you guys joining us after a long day of work. So the Hills webinar series is meant to bring you both clinical and non-clinical content that's relevant to your everyday life but rooted in nutrition. So that's really what we're trying to accomplish here, and we are so excited for Dr. McClay's webinar tonight because she is going to be speaking a lot about the science and the foundation for our new product. And so we are just absolutely thrilled and beyond excited about this product, and you guys will get the chance to listen to the master, the brains behind this entire operation. And I'm very honored to work with her on a mostly day basis, but here is her formal bio. Dr. McClay is the medical director at Hills Pet Nutrition, and she is boarded in internal medicine. Prior to joining Hills, she was an associate professor at Colorado State University. Dr. McClay received her DVM from The Ohio State University and PhD from the University of Minnesota in 1999. In her role with Hills, Dr. McClay has collaborated extensively on research that has transformed the lives of dogs and cats, including work on prevention of upper and lower urinary tract disease, obesity, dermatological disorders, and gastrointestinal disorders, which we have the distinct honor of hearing about tonight. So without further ado, Dr. McClay.
Thank you so much. I am very, very happy to have the opportunity to talk with you all this evening and this morning or this afternoon, depending on where in the world you're logging in from. We're going to have the opportunity today to talk a little bit about the gastrointestinal biome, and my goal is to get us all on the same page. We're going to start by a little bit of whimsy. These are our microbes. They've so far traveled the country with us. They've sat in on lectures and a lot of different webinars as we venture forth and start talking about the gastrointestinal microbiome. And so you two are going to join us today. So let's start by talking a little bit about what we're going to cover this evening or this morning or this afternoon. We're going to talk about how prebiotic fibers nourish the microbiome, and some more than others. That is to say that not all fiber sources that nourish the microbiome are the same. So we'll get to that a little bit once we cover some basic territory. The next thing that we're going to move on to is that the activated biome, and what I mean by that is when we nourish the microbiome appropriately, it becomes very active, and in so doing, it releases and produces microbial end products, which we call postbiotics, which have very potent biological activity. And then lastly, we're going to talk a little bit about how a fiber bundle that does support microbiome health can lead to rapid and long-lasting resolution of clinical signs, such as diarrhea. So I think we all start on the same plane in that we all understand that the intestinal microbiome is an ecosystem of organisms. So we tend to talk mostly about bacteria. There are other types of organisms that are compose the microbiome, but we're going to focus primarily on bacteria today. And there are lots of different ways that we can understand the microbiome in that context of an ecosystem. And we can use different kinds of analogies and metaphors, if you will. And I'm going to use one in particular today, and that is this idea of an orchard. And the reason for that is when we think of an orchard, we can think of the trees as being the microbes themselves, and they produce something. In this case, they produce, the microbes produce postbiotics, and in our metaphor, we're going to be talking about the fruit that is the product of an orchard. So the basic function of the gut microflora is that it harvests energy from food that bypasses digestion. And we're going to come back and redefine digestion just so that, again, we're all on that same page. And then those bacteria, of course, have byproducts. And those metabolites of their metabolism can exit the body, but they can also have an effect on the gut lining, and they can be absorbed into the circulation, and they can have profound effects on other organs as well. And it's really a key to health in that a healthy microbiome and how we feed it using advanced nutrition to nourish and activate it can have a very profound effect on health. Both in the short term and in the long term, and both locally within the intestinal tract and systemically as well. So a well-nourished microbiome produces postbiotics and releases bound compounds from dietary fiber. I've alluded to this once already. And essentially, prebiotic fibers are metabolized, and they can have trapped within them certain compounds called postbiotics. And those are really first-generation postbiotics, if you will. They're trapped in the fiber, and the only way that they're released and exposed to the body is when the bacteria come along and munch on them, if you will. You know, they metabolize those fiber sources for energy, and those trapped compounds are released. Once those trapped compounds are released, the bacteria may themselves further transform those compounds. They may metabolize them even further and produce even a second generation of postbiotics. So postbiotics can either be overtly released, or they can be released and then further transformed by the microbiome. And this is where some very unique fiber sources can provide some very potent effects on the body as well. The other thing that the microbiome does that's so important, and the postbiotics contribute to this as well, is that they help maintain the mucosal integrity of the intestinal tract. And they also contribute to a healthy immune system. And that's something that most of us are pretty familiar with. And then, of course, lastly, a nourished microbiome is an active, living population of microbes within the intestine. And as such, they consume excess protein, excess fat, and then, of course, those fiber sources as well to produce more bacteria. And that overall expands and increases the biomass within the gut. And that has benefits in and of itself. It encourages regular stooling behaviors. It encourages normal formed stools that aren't too hard and they aren't too soft. And in and of themselves, as we know, expansion within the gut stimulates motility. So there's a physical aspect to a healthy microbiome as well. Postbiotics, as I already mentioned, can act locally against the enterocytes and provide energy. The most familiar postbiotic that we all learn about in veterinary school are the short-chain fatty acids, so butyrate, propionate, that sort of thing. And those are actually used very locally for energy, but there's a whole host of other compounds that act as postbiotics. And many of these are absorbed into the body and can have an effect on the brain, so they can affect cognition. Some of them in human medicine have been linked to satiety or hunger drive. They can be linked to kidney health. So we've learned about these different compounds kind of en masse as a lot of the uremic toxins that may occur with kidney disease. They accumulate because in the circulation because they're not being cleared by the kidney. But the origin of many of those compounds is the gut microbiome, and then they're absorbed into the body. So postbiotics from fiber fermentation are generally can be very beneficial to the host. And you'll see if you do some reading that some definitions of postbiotics are only those things that are considered beneficial to the host, and others are more broad to say that any postbiotic that's produced by bacteria, whether it's good for the host or bad for the host, is still called a postbiotic. So right now, what we want you to take away from this lecture is just to understand what a postbiotic is, that it's the result of microbial action on prebiotic fibers. And of course, prebiotics differ from probiotics in that it's the nutrient, the nutritional source for the gut bacteria. When you give a pet or yourself a probiotic, what you're actually giving is living bacteria. Whereas, again, a prebiotic fiber is something that you consume. And the overall output of that the microbiome expansion, again, is bacterial protein synthesis. So a healthy gut bacteria microbiome makes more bacteria, and as I already mentioned, that also increases fecal bulk, and that has advantages. And those postbiotics can be used for energy, and they can also have benefits to the host. I mentioned some ones earlier around driving satiety. They can also be anti-inflammatory. They can be antioxidant. And that's really what we're going to focus on today because that's what we know the most about in dogs and cats. So here we're going to talk a little bit about what we feed bacteria. So up until now, everything I've talked about has been around this idea that we're feeding the bacteria a fiber source. And if you remember, fibers are really in that carbohydrate group, right? The complex carbohydrate, and fibers fall into the same category when we talk about proteins, fats, and carbohydrates. Fiber sources are a subset of that carbohydrate group. And in general, when we feed the microbiome fiber sources, they're very happy and they produce more healthful postbiotics. When they are fed, quote unquote, protein, that actually is a lot less healthful for the microbiome. And many of the postbiotics that accumulate in the body as a result of the microbiome eating a lot of protein can actually be very damaging to organs once they're absorbed. So when we have a disruption of the microbiome, the terms that can be applied to that would include dysbiosis or maladaptation. And when we have dysbiosis or maladaptation of the microbiome, this results in postbiotics that do not nourish the gut, and once absorbed into the body, can actually harm organs. And those disruptive events can be secondary to antibiotics, given sometimes we want to disrupt the microbiome if there's already a pathology present. So say we have an overgrowth of E. coli or Campylobacter or Salmonella. In those situations, sometimes we do want to use antibiotics, but we always have to use antibiotics with that understanding that it will disrupt the microbiome. It will always happen, and the goal is to have it happen for a reason and have the outcome that we desire. We now know that sometimes, in most species, when they're exposed to antibiotics, sometimes the result is a more long-standing disruption of the microbiome. Certain diseases, of course, can disrupt the microbiome as well, and then there can be simple dietary indiscretions, or, as I alluded to earlier, when we're feeding a total amount of protein to the dog or to the cat that's in excess of what they can digest, then that can also lead to dysbiosis or a maladaptation state. When that happens, what we see are postbiotics and byproducts of bacterial actions that are less desirable byproducts. So we see an accumulation of ammonia, which can change the pH of the intestinal lumen. We can see accumulation of phenols and indoles and sulfides. And again, all of those have an interaction with other bacteria. And so, quote-unquote, good bacteria may decline within the gut, and bacteria that prefer to eat those protein sources for energy become more abundant, and then as a result of that, what we see are postbiotics that generally act as being more pro-inflammatory, again, both locally and systemically. And of course, we see less energy for enterocytes because we're producing less butyrate and propionate. So, as I promised you earlier, I said that we would talk a little bit about digestion. We often use that term, and we generally think of digestion as everything from the mouth all the way to the anus. But technically, that's really not true. And in dogs and cats, most digestion occurs in the stomach and the small intestine. Very, very little true digestion takes place in the colon. What happens there is the action of the microbes, and so what we really have is lytic actions. So the microbes lyse those fiber sources. That's called saccharolysis. And then we also see proteolysis that can occur in the colon as well if there's protein sources that are entering. If we have an excessive amount of fat, and that's also making it through the small intestine, then we'll see that present to the colon as well. And so proteins, fats, and starches are truly digested and absorbed in the small intestine. And if you think about how we typically feed a dog or a cat, ideally what we want to do is feed protein and simple starches and fats so that they are as close to completely digested in the small intestine as possible, and so that there's very little spillover into the colon, because as we've already talked about, the colon is really happiest when we are feeding it fiber so it can undergo saccharolysis and fermentation. And the amount of protein that those bacteria see is relatively low, and it's really just enough so that they can make more bacteria and not so much that they're consuming the protein for energy. We often think of the fact that only fiber sources can be fermented, but in actuality, protein sources can be fermented as well. So let's come back to our metaphor that I mentioned earlier. If we think of nutrients that we're feeding our microbiome, like the water and fertilizers that we might use in our orchard, then those bacteria producing postbiotics are like those trees. And if they're well fed and well watered, then we're going to have a nice bountiful harvest. We're going to have lots and lots of very healthy postbiotics. And that's really what we want. We want those bacteria to produce those really good postbiotics. And we tend to think that bacteria are like those trees, and some bacteria truly are. They're what we call specialists. And specialists are like the apple tree that we have here. So specialists really prefer to consume only one kind of nutrient source. Specialists may prefer protein, or they may prefer fiber. And as we've talked about before, the types of bacteria that prefer to consume fiber produce more beneficial postbiotics than the types of bacteria that like to consume protein. And so those are the types of bacteria that we are encouraging to grow when we feed foods that are rich in fiber sources. And that occurs obviously for us as well. However, this is where our metaphor breaks down a little bit, because most bacteria, the majority of bacteria in the gut, are actually generalists. And that means they'll ferment whatever they can get their hands on, so to speak. So they may prefer to ferment fiber over protein. However, if they're not getting much fiber and they're only getting protein, that's what they will consume. And so the postbiotics that they produce are dependent upon what they are fed. And so it's almost like a magic tree. It could produce apples or pears or oranges depending on what those particular bacteria are fed. And so we'll see different postbiotics. And this is where some unique fiber sources can really come into play, because we might get some very special postbiotics if we feed them to our generalists that we might not necessarily see otherwise. This also gives us an advantage in that when we talk about the microbiome, you'll often hear that every pet's microbiome is unique. And then you'll hear that we can influence the microbiome, say, with a particular food or a particular nutrient source. And how do we kind of reconcile those two things? And the reason why we can, again, is because many of those bacteria are generalists. And so when we talk about a pet's microbiome being unique, which talks to the exact numbers of different taxonomies of bacteria, in reality, many of those different taxonomies of bacteria function very similarly. And so if you can have a pretty healthy gut microbiome, actually with very little taxonomic diversity, because you actually have bacteria there that are very functionally diverse. You have a lot of generalists. And that's where, if you hear somebody talking about a microbiome being diverse or not being diverse, you need to dive in a little bit more closely to figure out exactly what they're talking about. Because you could have a pathologic state that has very low diversity, say an animal that has a chronic overgrowth of salmonella or E. coli. So there's a pathogenic bacteria that's dominant. Or you can have a very, very healthy animal that has a low amount of taxonomic diversity, but they're very, very healthy because the bacteria that are there are very functionally diverse and you're feeding them well. And so those bacteria can really serve that pet well. So let's talk about the idea that fiber sources are not all the same. As I mentioned earlier, they aren't, and this is just an example of 26 different fiber sources, which I have here across the bottom. And what we can see is I just picked one postbiotic. In this case, we picked butyrate. And what you can tell is in the amount of butyrate that can be produced, and this is historical data over the years looking at, again, 26 different fiber sources here at Hills, about a 30-fold difference between the ability to produce butyrate from the first fiber source compared to this 26th fiber source. And that is absolutely true for all different fiber sources. That is, there's a lot of diversity in the different compounds that they produce. And when you combine that with the fact that there are hundreds of different postbiotic compounds that we can look at, you could see that a lot of different fiber sources may have a lot of different potential benefits depending on how they're used, how much of them is used, and the total formulation of a particular food. And so we here at Hills are gaining quite a bit of information and knowledge around these different fiber sources. And what is so fascinating and so exciting about this is that we know that for so long people have thought about fiber as being very inert, right? We just think about it as something that helps normalize water within the gut. It helps with stooling behavior. And if you talk to some pet owners, they may say, oh, you know, fiber is really just there to make my pet feel full. It's used by pet food companies as a filler so that they don't have to put other ingredients in there. And that's really not true. Fiber has a lot of biological function, and by understanding what ingredients we have at our disposal, we can really build a food around the end products that we're looking for depending on the vision for that particular food. So for example, if we are talking about energy, so something like short-chain fatty acids, butyrate, propionate, that sort of thing, we might choose foods that are very good at producing those things. So we find those in a lot of different prebiotic fiber sources. To use a specific example, flaxseed is one, and flaxseed actually produces in the healthy microbiome quite a bit of enterodiol, and enterodiol can have very potent anti-inflammatory activity. If we look at different plant-derived polyphenols that can be trapped within plant-derived fiber sources, we can see a whole host of different compounds. And just two examples here are ones that words we're not really very familiar with, such as hesperetin and 4-hydroxycinnamate. These are just two examples of those different compounds. And these two in particular have antioxidant activities. So let's sum up a little bit about what we've learned so far. So we've talked about the fact that within the colon, bacteria use fiber to produce postbiotics which act locally and systemically. So we can have different prebiotic and fiber sources that are derived from different plant compounds. They can undergo saccharolysis and release of those fiber-bound plant compounds by the bacteria. Then they can be broken down into monosaccharides, and those monosaccharides can undergo fermentation. And the products of that fermentation include different postbiotics, short-chain fatty acids such as butyrate, acetate, and propionate, and those can have both local activity on the enterocytes and be absorbed into the intestine. Further, we know that those plant compounds can be broken down and transformed into those first and second generation postbiotics that we talked about, and those may also undergo further transformation to produce those second generation type ones. And so they can also have local and systemic effects. And those are often they can be antioxidant and anti-inflammatory compounds, and probably the ones that we're most familiar with are polyphenols. So, up until now, we've talked about this idea of how do we nourish and encourage growth of desirable bacteria. And we can do that through use of fiber sources. We can also activate the bacteria that are there by feeding them special fiber sources that can produce first and second generation postbiotics. And then lastly, what we're going to do is we're going to talk about how a specific fiber bundle that we've researched here at Hill's Pet Nutrition can actually lead to real rapid and long-lasting clinical results, because that's what you're really interested in. So the basic science is really interesting, but let's put it into action. So we first identified a combination of fiber sources that we were particularly interested in, given all of our basic work. About 5 years ago, we really started an aggressive research program here at the Pet Nutrition Center to develop our microbiome capabilities. Now that big data was really at our fingertips, because you can imagine that there's quite a bit of data that you need to understand and deal with when you're developing foods specifically to enhance the activity of the microbiome. And we started to look at how feeding balanced foods with a fiber bundle that we named Active Biome Plus, how it could nourish and encourage saccharolytic bacteria. We looked at different sources so that we could see if we were liberating those hidden plant compounds from within the fiber matrix, and whether or not those hidden compounds were being further metabolized and transformed by the gut bacteria to produce those second generation biotics. So we'll talk about both dogs and cats, but let's start with some cats. We had an 8-week study where we fed 46 healthy cats first a control food and then a gastrointestinal biome food, and we examined those cats after 1 and 2 months of feeding. In this particular plot, called a radar plot, what we can see are these ideas of, are we nourishing the microbiome? Are they producing these saccharolytic monosaccharides that can be used to directly create energy within the gut? And so what you see here in the center in red is the control food. And then what you see in the purple is after 1 month, and after the green is after 2 months. Each circle on the radar plot represents a log difference in the production of that particular compound. And so what we can take from this, here we have the red, here we have the purple and the green, and again, each one of these rings is a log difference in the production. And so what you can see is in some situations, such as here, we have many fold differences in arabinose between the two foods. Well, not quite so much of a difference between baseline and the food for something like the acetoheptulose. And this is not an exhaustive list around the outside of these different compounds. We've picked ones for illustrative purposes. In this radar plot, what we're showing you is not only has that microbiome been fed, we're showing those byproducts of being fed in the first radar plot on the previous slide. Now we're showing that beneficial plant compounds from the fiber sources that are in the food are actually being released. And so again, when the cats were consuming the control food, we have this inner red area for each of these different compounds. And the purple and the green are almost exactly overlaid with each other. And so this is one we mentioned early, hesperetin, you can see has a many-fold difference compared to the control group. The last thing we talked about is, as far as the activated microbiome, is, are those liberated compounds being further transformed by the gut microbiome? And we can definitely see that here. So here's an example. Again, in the center we have the control food. Each ring is a fold difference, a log difference between the other. And again, the 1 month and the 2 month data are pretty much sitting right on top of each other. But you can see for a compound such as enterodiol, a many fold difference between the control state and the GI biome fed state. So what does this tell us? It tells us two things. It tells us that we are, in fact, nourishing and activating the microbiome, which is the goal. And the second thing is, people often will say that cats are different because their intestinal tract is generally shorter, because of the fact that they are obligate carnivores, that they cannot metabolize and take advantage of microbiome and fiber benefits the same as dogs or humans potentially can. And the truth of that is that is not true. Cats can. We just showed very good data to show that they do, in fact, can metabolize fiber sources and release all of those beneficial compounds. So again, the right prebiotic fibers can, in fact, nourish and activate the microbiome. So the next thing we're going to talk about is some clinical study data. The previous work that I showed you was in healthy cats, and we had similar data in healthy dogs. In the interest of time, we're going to move on, and I'm going to show you data from dogs that had diarrhea. So in this clinical study, it was a prospective single treatment arm study, and the reason why it's a single treatment arm is because it's really not appropriate not to treat dogs with diarrhea, and so we couldn't have a negative control. Dogs that were included in this particular study were 1 to 10 years of age, and they had either a current episode of diarrhea that had persisted for at least 2 weeks, and that included if they had had prior therapy, or if they had a history of chronic persistent diarrhea that had previous episodes that had lasted for at least 2 weeks and were now presenting with a new current bout of diarrhea. We, of course, excluded dogs that had parasites, evidence of concurrent diseases, dogs that predominantly lived outdoors, which meant that the pet parent couldn't observe how the treatments were going. If they were on therapeutic foods for some other reason, which often means they have a concurrent disease. If they were on oral antibiotics for any other reason, or for diarrhea this time around, or meds that might influence the motility of the intestine. And this slide shows you the study conduct. So essentially, these are the different actions that occurred throughout the study, and what I want to bring your attention to is the day of the study. So what we have here is day 1, day 2, and day 3, and then 2 weeks, 4 weeks, and 8 weeks. And so for these first 3 days, the dogs stayed in the clinic. And that was so that they could be closely observed for how well they were responding, and so that fecal collections and blood collections could take place as well. And most owners were more than happy to leave their pet with diarrhea so they wouldn't have to be cleaning up after it for these first few days. We enrolled 31 dogs, and 22 completed the study. The reason why we had 9 dogs leave the study, 1 dog for unrelated medical conditions. We had several dogs that didn't want to eat within the while being boarded at the veterinary hospital, and we didn't want to prevent those pets from eating, and so they went home because they were happier to eat at home. And the average age was about 5.5 years. We had a great mix of different breeds of dogs. So we had 11 mixed breeds, and terriers, retrievers, beagles. You can see the list yourself. We had a very nice mix of both male and female dogs, and all dogs, again, had been diagnosed with chronic enterocolitis. The vast majority had mixed small and large animal signs. Then the next majority had large colon signs, and we only had a few dogs, maybe 2, I think, that had exclusively small intestinal signs. At entry, the mean stool score was 2.6 out of 5, which is not a big surprise because they had to have diarrhea in order to be admitted into the study. And for those of you who aren't familiar with our grading system, I know that some grading systems go the opposite, so low numbers are better or more firm. In our particular case, a higher number represents a more firm stool. So I'm not going to dwell too much on this because we talked about it in the previous work where I was sharing you the work in healthy cats. What we were able to confirm here in these dogs with underlying diarrhea is we saw much the same thing. So when we looked to see whether or not we had byproducts of saccharolysis and energy metabolism, we saw that. So we were nourishing the microbiome even in these dogs that had diarrhea. And then we saw first and second generation postbiotic compounds. So compounds were being liberated from the fiber sources, and some of them were being further metabolized into new plant compounds. So we were both nourishing and activating the microbiome. what you're probably most interested in is how the dogs did. So we asked the veterinarians whether or not the dog was responding or not, and they could answer that the dog had a complete response, a positive response, was not responding, had a negative response, or actually had a recurrence of diarrhea. And what you're seeing here is day 2, 3, 4, so these first 3 days, and then 4 weeks and 8 weeks. And what you can see is there's no black, so there's no recurrence in any of those times. And by day 4, we had almost 100%. By 4 weeks, we had 100% of those dogs had either a positive or a complete response, and that remained steady throughout to the end of the study. So let's look at that with respect to the stool scores. What we saw were that stool scores improved significantly in that first day. So they went from 2.6 to 3.8, and that was a statistically significant change. They further improved within those first few days. This is day 5, and what we saw was that by then the average stool scores were over 4.5, and they remained that way for the entire duration of the study, and that was for all dogs. And so the average for all dogs remained very high throughout the study. We had 100% of the dogs had a positive response. So what we've covered today is a little bit around some definitions. We've reviewed this idea of digestion and reminded ourselves that that really occurs in the stomach and small intestine. And so when we're talking about the microbiome, it's really around saccharolysis and making saccharolysis much more prominent compared to proteolysis. And we can do that by feeding prebiotic fibers. And then if we want further benefit, we can look to very specific fiber sources that produce very healthful postbiotics, such as polyphenols. And those polyphenols can be absorbed and act locally. They can also be further transformed into new compounds by the gut, and those can also have beneficial effects both locally and systemically. And those effects primarily are in the areas of being anti-inflammatory as well as being antioxidant. And so what does that mean clinically? It means that in dogs with chronic diarrhea, as in that we saw in this particular case, you can have very rapid changes in the gut that can result in resolution of diarrhea in as little as 24 hours. And that those benefits can be maintained for an extended period of time. So with that, I would be more than happy to answer some questions.
Thank you very much for that, Dr. McClay. That was a really good introduction. So I guess the big question, so this is a new product that's developing, is the Active Biome going to be a product on its own, in its own special food, or is it going to be something that is going to be incorporated into some existing
So gastrointestinal biome is the name of the food, and it will have the Active Biome Plus fiber bundle in it. And right now, that is the place that you will find this particular fiber bundle within our portfolio. And our vision for this particular product is for those dogs that I talked about, those chronically affected dogs that have recurrent or almost fairly consistent enterocolitis signs. It can also be that these are dogs that have chronically loose stools or soft stools. One of the things that we have found is, it's not uncommon for us as veterinarians to focus in on pathology. So we'll ask a pet parent, does your dog have diarrhea? And the pet parent will say, no, they don't have diarrhea. But if you say, does your dog have soft stool or stool that's difficult to pick up and kind of mushy, very often they will say, well, yeah, they have that, but it comes and goes, or it's not so bad. He just runs out in the backyard, and so I don't have to pick it up, so it's not that big a deal. Whereas in other parts of the world, if you have an urban dog, that's a much bigger problem. And so, but both of those scenarios, that dog probably has either some dysbiosis or some maladjustment that's happening in the gut microbiome. And I think a lot of those dogs absolutely will benefit from this food as well.
Fantastic. Now we've got a couple of questions about the source of the fibers, but before I get to that, we mentioned the biome there and dysbiosis. Are there any plans for any future trials looking at the use of probiotics, either in conjunction or as a treatment control?
Yeah, and I think that's a fabulous and a very interesting question. It really does add a lot of complexity to study design. It can be very difficult to study probiotics on their own and fiber sources on their own, and then to combine it and look at identifying cases that are pretty consistent so that we can eliminate a lot of variability so that you can do a really good, thorough, rigorous scientific investigation can be a challenge. The other thing out there is that many probiotics are not species specific, and so we know that their activity within the gut may be short-lived, so it's only really seen while it's being actively given. And our philosophy is really to feed the gut microbiome that's present in these particular dogs. Yeah.
Work with it.
No. I mean, I'm not saying that probiotics necessarily are against it, not to put words in your mouth, but I think, yeah, because I don't want to give that impression. It's just when some probiotics don't have a lot of bacteria, some do have a lot of bacteria, they may or may not have a tremendously rich, diverse number of bacteria. So one of the topic questions that often comes up is really, it's more, the gold standard for a probiotic is really a fecal transplant, right? So you're giving a large quantity of bacteria, and you're giving really the full spectrum of taxonomy that might be there. And so certainly, again, there might be a place, if you're going to do a fecal transplant, by all means, you want to back that up by feeding the bacteria that you're giving that particular pet.
Exactly. Now, Mel and Dolores have both asked a bit about the ingredients that go into this Active Biome Plus product. So, what are the sources of these prebiotic fibers?
Well, there are many, and so thank you for asking the question, both Mel and Dolores. In this particular product, we're looking at citrus pulp is one, and so we know that there is a tremendous amount of good polyphenols and actives in citrus pulp, and cranberry pumice as well. Both of those carry with them quite a bit of polyphenols that are in them. If you look at them, and I know you can't see through the computer, but if you were looking at it, at say cranberry ingredient, it actually is red. So even though the juice has been squeezed out of it, there's still a tremendous number of polyphenol type actives that are still in that as a fiber source. And other ones include flaxseed, which we mentioned, beet pulp, which is another, and then the most unique one is pecan shell fiber. And most people kind of look at us strangely when we start talking about pecan shell fiber, but there are actually some amazing polyphenols that are trapped within the lignin of pecan shells. And pecan shells are completely devoid of any nut proteins, and they are, again, have these special lignins with a lot of polyphenols entrapped within them. And so when it's ground into a powder, it's actually a very nutritious fiber source for the microbiome.
Well, there you go. Who'd have thought? Who'd have thought? Now, we've got a couple of questions about other possible indications for this, I guess, technology. So Joanne has asked about bringing feces not from the soft to the firm, but possibly for dysbioses or problems with digestion that are making the feces too firm. Is there any role, do you think, for using these postbiotics for bringing
I think
food back to normal?
absolutely. So the most common scenario there is cats, and we do talk about gastrointestinal biome as normalizing feces, good healthy stool in cats that have a history of constipation, as well as cats that may be firm, and the same for dogs, for certainly. Fiber is really somewhat, I hate to use the word magical, but it really has wonderful water holding capabilities. And so there's several things in play, right? So we know that fiber can hold water, so if feces is too loose, it tends to give up some of that. If it's too firm, it tends to draw some in. And then we also know that an inflamed intestine tends to be more hypersecretory, or it can be dry. And so when we have poor motility, sometimes we'll see drier feces. And so it's that multiple facets that we see of a healthy microbiome having biomass, a healthy microbiome producing these anti-inflammatory compounds, and then the physical attributes of holding and releasing water all contribute to healthy intestinal and fecal production.
Sounds like there's a lot more for us to learn about this, and it's a fantastic area to be working in.
Yeah, it's a lot more than just a filler, isn't it?
Yeah, exactly. And it's just such an exciting opportunity. We've got a couple more minutes for some last-minute questions. So might do them rapid fire so we can get everyone in. But we've got some fantastic engagement from the audience, so that's always a really good sign. Blake has asked a question about using this as a diet to support patients who are being treated with antibiotics to help support the gut through that process. Is that something that would be looked at as a label claim? Obviously it's a new product, but do you think that there's an effect when we are dealing with something that's going to stress out the gut microbiome, whether that is a course of medication or anything like that?
Yeah, I think that's a wonderful question. And so what I can say is we don't have any data specifically around the use of this particular product with or without antibiotics. However, what we would say is that every case is unique. We all practice the art of medicine, and so we have learned that there's an impact of antibiotics on the gut microbiome. Could this help in some of those cases? I think there's a possibility there. However, again, we don't have data, and you need to treat your patients. So I don't think we would ever say don't use antibiotics and use this food, because that's not appropriate in the same way. So our hope is that we're going to support the patient, we're going to support their microbiome so that you can practice the best medicine that you can practice.
Fantastic. And then thinking about the future, I mean, everyone's very forward-minded, which is always great to have in a presentation like this. The role of postbiotics and the microbiome in other conditions, such as dermatological conditions and other immune-mediated diseases. What's the future look like for postbiotics?
I think the future looks bright for humans as well as pets. So there's so much that we're learning about the microbiome every day. We know that the skin has its own innate microbiome. We know, obviously, that the gut does. We know that the urogenital tract does. And so there's so much that we can learn as we move forward. And how all of these different microbiomes interact is a great area of research. And so the future is very bright. I don't know exactly where it will lead. I wish I did. I'd probably be famous if I could predict the future, but there's so much that we're learning every day.
Well, on that note, I'd like to thank you for sharing with us tonight. And I'd like to thank everyone for signing in, and I've gotten a lot out of this, and I'm sure everyone else has. Now, that was packed with content, so I'd like to thank Dr. McClay. Thank you very much for joining us, and enjoy the music as we sign out. Thanks very much.